In this video, we will discuss about the without membrane organelle of a cell. Different without membrane organelles present in a cell are ribosomes and centrosome. These ribosomes are the smallest organelle present in a cell. These are the granular structures which are present in both prokaryotic as well as eukaryotic cell. In case of prokaryotic cell, these are 70S type of ribosomes, and in eukaryotic cell, here 80S type of ribosomes present. These ribosomes were discovered by George Faraday. In 1955. So these are also called as Faraday body. Now we see that these ribosomes, of which material these ribosomes are made up of. These ribosomes are made up of rRNA and proteins. Now question which comes in our mind, that how a without membrane organelle exists. Suppose you can see here, suppose this is a protein, polypeptide or a protein chain, and this is a RNA molecule, and these molecules just folded one upon the another in a 3D or tertiary structure. So there is no need of a membrane which enclose this. Similar to this, both these proteins and rRNA are folded upon one upon another and form a structure that is called as ribosome. Now we will see different type of ribosome. In case of prokaryotes, here 70S type of ribosomes are there. Here the term S means sweat gall unit. Here 70S type of ribosome. This 70S type of ribosome exists in two parts, the smaller subunit and larger subunit. This is smaller subunit and larger subunit. This smaller subunit is made up of that, and this smaller subunit is called as 30S and larger subunit is 50S. And this 30S subunit is made up of 16S rRNA plus 21 type of proteins. While this larger subunit is made up of 23S rRNA plus 5S rRNA and 30 or 34 type of proteins. As this smaller subunit and larger subunit exist in the cytoplasm in dissociated form. At the time of protein synthesis, both these smaller subunit and larger subunit bind at a mRNA molecule and exist as associated form. In case of eukaryotes, there is 80S type of ribosome, and this 80S type of ribosome is again made up of two subunits, smaller subunit and larger subunit. This smaller subunit here is 40S and larger subunit is 60S. This smaller subunit in 80S ribosome is made up of 18S rRNA plus 30 subunit bind at the mRNA molecule. A large number of ribosomes bind at a single mRNA molecule. This is mRNA and this one is ribosome. So this structure which is formed by the binding of a large number of ribosomes on the single mRNA, this is called as polysome or polyribosome. So this polysome or polyribosome structure are formed in the cells which are actively engaged in protein synthesis. So these ribosomes can be called as protein factory because these help in the protein synthesis. Also these protect the protein molecule from the enzymes, protease enzymes, and also protect the mRNA molecule from the nuclease enzymes. So these are the main functions of the ribosomes, that they help in the protein synthesis and they stabilize the or help in the survival of the protein as well as the stability of the mRNA. The second without membrane organelle is centrosome. It is also without membrane and present in animal cells, not in plant cell. This structure is present in animal cells only and help in cell division. Here in the centrosome, the two centrioles, just like these are two cylindrical structures, centrioles, which arrange themselves perpendicular to each other like this. Here two cylindrical structures, these are centriole. This centriole has membrane, but these two centrioles which are perpendicular to each other are arranged and the dense cytoplasm also surrounding it. This complete structure, two centriole along with the dense cytoplasm, this is called as centrosome. So here this centrosome does not have any membrane, while these centrioles have membrane. So this centrosome help in the cell division of the animal cell. This is also a without membrane organelle. Now these ribosomes help in protein synthesis